Ph.D. candidate in the School of Biological Sciences at Washington State University in Pullman, Washington. For her Ph.D., Emily studies the ecology of black-legged ticks, an ectoparasite responsible for transmitting the causative agents of Lyme disease, anaplasmosis, babesiosis, and powassan encephalitis. Her work focuses on how free-living black-legged ticks end up on their various host species, and if this is driven by chance encounters between host and tick, or tick preference towards high-quality hosts. Prior to starting her PhD, Emily graduated with a BS in biology from Villanova University of Pennsylvania, where she studied hybridizing chickadees.
Yeah. So I can talk about very specific hybridizing chickadees. Cool. Ticks! We love ticks! Alright. So I'm going to start with like a little bit of background about myself and how I got into studying ticks, because a lot of times people are like, why would you study ticks? And then we'll talk about the background from my research about how we figure out how ticks select a host, and then the project that I've been working on here, as well as at the Carey Institute of Ecosystem Studies in Millbrook. To start, I'm not from Washington. I'm originally from southern Maine. I grew up in Portland, Maine, which is southern coastal Maine. Really nice. Highly recommended for a vacation if you haven't been before. For college, I went to Villanova University, which is outside of Philadelphia. If you follow NCAA basketball, I was there while they won 2 national championships. It was very exciting. I originally started at Villanova as a biology major with the intention of going to medical school, and quickly realized I did not like that path for myself. It just wasn't for me for a lot of different reasons, but I stayed with the biology major, and I just was trying to figure it out. So I was taking all my different classes, and I took an ecology course that I really liked. And ecology is the study of how organisms interact with each other and their environment. During this ecology course, we had one lecture, and I don't even remember if it was like one full lecture or just like a part of one lecture, about disease ecology. So it was ecology, so interactions between organisms and their environment as it relates to diseases. So here we have a picture that's just trying to get into that schematic of how pathogens can spill over between different host species through different mechanisms, including a vector, like a mosquito here, and how that can then also get into human populations, and how all these things are connected and influence our likelihood of getting certain diseases. And I was really interested in this idea of spillover of infectious agents from wild populations into humans, and I would always ask people, like, what's the next big one going to be?
Yeah, like the Wuhan market.
And yeah, so like, fun fact, people have known about that for decades now, that was, situations like that are going to be an issue. But there's a lot of really cool disease ecology research. So, as one should do when you get interested in something in undergrad, I tried to find a summer job that would get me more experience in the field. And so I ended up getting this research position at the Maine Medical Center Vector-Borne Disease Lab, where I studied black-legged ticks. So black-legged ticks are a common vector of disease in the Northeast. Here we have their different life stages. So we have larvae, which are the youngest of the black-legged tick life stages. These are ones that are hatching from eggs. And they hatch usually around like late summer, so they should be coming out in a few weeks. The larvae will be coming out. Nymphs are the next life stage, so after a larva gets a blood meal, it will molt and come out the next spring as a nymph. This season we kind of just exited. They're coming out around like May, June, into July a bit. And then the adults here are the big ones, and the adult female is what we typically associate with the black-legged tick, or you may have heard them called deer ticks, but black-legged tick is correct term since they feed on more than just deer. The males, they're kind of a little bit smaller than the females, and they're not as cool because they're not red and typical drought animals. But they're really cool. Particularly interesting for human health is the nymphal life stage, because they're the ones that are active while humans are active outside. So those are the ones that you should look out for. As a little handout, because there were handouts last week and I thought I should have some handouts, I have ticks in they're dead, they won't hurt you. That's fine. So here I have black-legged tick larva, so the smallest one, and this, I'm sure people have seen ticks before, but it gives you an idea of like really what size they are. Here we have some nymphs. Over here, I'm going to do adult female black-legged ticks, start in the back corner. And then I actually have an adult dog tick as well. This is the other common tick species you'll see in the Northeast. I'm not going to talk about them today because they're not as cool as black-legged ticks, but they exist and it's worth knowing what they look like.
Do they all cause disease or just some? I mean, female, male, and...
They can all potentially transmit disease. Larvae are way less likely. Most of the pathogens you know about, ticks are not born with. They're not transmitted vertically from mother to larval tick. So larval ticks have to feed on the host, get a pathogen from the host, and then the nymphs become infected. Adults are most likely to be infected because they've fed on more hosts than other ticks, so they've had more opportunity. But they're most active later in the year when people are less likely to be outside, or when they are active, they're a lot bigger than nymphs, so you're going to notice them on you more than you would for a nymph, or the adults will notice them. They're about the size of a poppy seed. The CDC let out a really horrifying image last year, a couple years ago, where it was a lemon poppy seed muffin, and they're like, "Stop the ticks." And people couldn't. It was scary to them. Alright, here we go. Slide. Okay, so black-legged ticks, they're medically important vectors of disease. A vector is basically an organism that transmits pathogens between hosts, usually arthropods, so things like mosquitoes and fleas and ticks. So pathogens of concern in the United States that ticks can transmit include the Lyme disease pathogen, Borrelia burgdorferi, what causes anaplasmosis, so Anaplasma phagoscopulum, the parasite that causes babesiosis, and Powassan virus. Powassan virus is something that's in the news. It's not as common, but it's like the really scary one that's out there that is pretty fatal. So don't get that one. Don't get it. Something to note about all of these tick-borne diseases, but we track it best in Lyme disease, is that they have been increasing in case numbers, so the number of people in the country or in North America in general that are getting Lyme disease is increasing. It's also increasing geographic spread. So here we have the number of reported Lyme disease cases on a map from 2001. You can see it's really concentrated on the Northeast, but not super far up north yet, and also kind of in the Great Lakes area. About 20 years later, it's expanded way more inland. You can see that it's now reaching almost the entire state of Pennsylvania and New York. Massachusetts does have a lot of ticks. It was just a reporting issue this year. Don't think that there's some sort of like magical situation happening in Massachusetts. They have plenty of ticks. It's also expanding more in the Great Lakes region. And looking at just Lyme disease, there's about 300,000 cases annually, which costs the U.S. healthcare system somewhere between $700 million and $1.3 billion annually. So it's a big problem. It's a big, big problem. And it's, I think, the most common vector-borne disease in the temperate world, so temperate North America. So I studied ticks for a summer internship. I was like, this is so cool. I was really interested in how things were changing and like how the ticks interact with their environment and how they got pathogens. And so I did what you should do if you're really interested in studying something, which is I applied to grad school. So I ended up going to Washington State University, which is in eastern Washington on the border with Idaho, in this region called the Blues, which is these rolling hills. It's very agricultural. And it's really interesting looking out there. I'd never seen anything like it. But if you recall from the previous slide, ticks and tick-borne diseases seem to be more on the East Coast, and then I moved out to the West Coast, but I'm trying to study ticks ecology and the environment, which means I have to come back out to the East Coast to study ticks for my fieldwork. So for my Ph.D., which I started in 2018, just about every summer, with the exception of 2020, because we all know why, I've been coming out to New York to the Carey Institute of Ecosystem Studies. And some people might be familiar with the Carey Institutes of Global Good down the road. Carey Institute of Ecosystem Studies is different. A lot of the tick-borne research that you see about the state of New York is actually coming out of a lab at the Carey Institute that I work out of. So it's really nice to have this established tick research program where they've been studying tick-borne diseases as it relates to environmental fluctuations and tree seed production, mammal populations, and all sorts of things. So they have really good infrastructure for me to just show up and do my own work. All right, so this is how I got to be studying tick-borne diseases. Now we're going to talk about parasites a bit more generally, and then we can get into my research. So parasites are creatures that have a really close relationship with another animal, their host, and they use their host for food and, I guess, shelter, essentially. And parasites exist on the spectrum from specialist to generalist. So a specialist parasite, parasites use only one or very few host species. So here we have a louse. I think I should stand over here or something. Here we have a louse that only feeds on this one species of pigeon. So this would be a specialist. You can have a couple host species and still be considered a specialist. A generalist would be something like our black-legged tick, which can feed on, I think there's recorded associations with over 125 host species, including small mammals like mice, larger mammals like deer, birds, and also reptiles. Isn't that fun? It seems like they try to feed on anything. And so the maps I showed earlier on disease cases, ticks actually expand all the way down the eastern seaboard, and further south they're more associated with lizards than they are with mammals. Interesting, right?
So they actually hop from those animals onto us?
So ticks spend most of their life just in the environment. They live for about like a year and a half or so, and they only feed on 3 hosts in their entire life. And so in the time between their feeding on hosts, which they'll feed on a host for a couple days and then drop off, they just exist in the environment. So if a tick gets onto you, that means that you were its attempt at finding a host. Because they'll potentially try to feed on anything, I guess. Maybe. We'll see. We'll see where that goes. So many generalist parasites, so parasites that use a whole bunch of species, they don't necessarily use all those host species equally. They use them in a consistently uneven pattern. So here's a figure that tries to get at that distinction. So basic specificity is what I was just talking about with the straight up just number of hosts they're using. So a parasite with low specificity would use a bunch of hosts. It's not very specific. It's just, it has all these different host species. With high basic specificity, it uses fewer. With something like structural specificity, we're looking at the quantitative distribution of parasites on hosts. So here, it's still using the same number of hosts, but you can see that it's predominantly using one host species over the other. So this is like saying, like, I have a generalist diet, I'll eat a lot of things, but in reality, every day I'm eating pasta. You know, like, am I actually getting a varied diet? No, I could, but I'm not. You know, so that's what some parasites are doing. And when we think about the consequences of being generalist or specialist, sometimes we just think about this flat number of hosts that a parasite could use, and we don't think about how they're actually using their hosts. So with that in mind, the black-legged tick and Lyme disease system is highly likely to be impacted by uneven feeding. So if they're not using all those 125 potential hosts evenly. So they're generalists, over 125 host species. They feed on one host per life stage. So a larval blood meal, they drop off, they molt, they try to survive the winter, they emerge as a nymph, they try to find a host, they feed, they drop off, they molt, then they come out as an adult, they feed, they reproduce, lay eggs and die. That's the cycle of a tick. So fun. And for the most part, they can only acquire pathogens like Lyme disease by feeding on infected hosts. So larval tick is born uninfected, feeds on an infected host, becomes an infected nymph. That infected nymph will feed on a mouse and make it infected. This cycle continues. Now, something to note, they feed on all these different host species. Maybe they feed on them evenly, maybe they don't. Host species vary greatly in their competence, which is this term describing how well a host is able to harbor and transmit pathogens to vectors. So in the case of Lyme disease, a host like a mouse, and I should have put like little animal pictures to make it easier to see. So a host like a mouse, we have a mouse here, has a really high reservoir competence. So a mouse that is infected with Lyme disease is really likely to pass it on to ticks. So when a tick feeds on a mouse, it has a really good chance of getting Lyme disease. Hosts like opossums, which we were talking about, are less good hosts for things like Lyme disease. So if a tick feeds on an opossum, it's way less likely to get a pathogen. So maybe should we just like try to force all the ticks to feed on opossums? I don't know. Maybe. Yes. Something else to keep in mind. So that's the quality for the pathogens. This is hosts vary in quality for ticks themselves too. So not every host is equal for a tick. So hosts vary in feeding success for ticks. So this is basically a measurement of if a tick can get a full blood meal, if it's able to survive the couple days it needs on a host to get a full blood meal. If after it feeds, it successfully molts into the next life stage, and this is in particular looking at the transition from larval tick feeding on a host to nymph, the likelihood it survives the winter after molting, and then combined, that's the overall survival of a larval tick feeding on a host species to becoming a nymph. So this first row is after feeding on a white-footed mouse. They have a relatively high feeding success rate, pretty good at molting after. Overwintering is okay, and so their overall probability of surviving to becoming a nymph is decent in the range of what could happen. Now take our friends the opossums. Ticks are way more likely to die feeding on an opossum than successfully feed.
Why?
I don't know. I can't tell you. The hypothesis is that opossums are maybe better groomers than mice are. They do kind of clean themselves. I know they seem really stinky and gross, but they do kind of fastidiously clean themselves pretty well. They also might just have thicker skin, and it's harder for the ticks to penetrate the fur to get to the skin to feed. And then their overwintering survival is lower. My guess is that those that do feed maybe don't get a really good blood meal, so they don't have as much nutrition to survive the later stages, but I can't confirm that with data. So yeah, ticks that try to feed on opossums, they were really lucky if they survived to the next life stage. And so with this wide range of permissiveness and the likelihood that you die if you feed on a live host, you would expect ticks to specialize to avoid bad hosts. Like if you were a tick, you would avoid an opossum, probably, presumably, right? Like you're not going to go out in the woods and try to eat poison ivy and be like, oh, maybe it's fine. So we would expect them to specialize on permissive hosts and avoid non-permissive hosts. And in reality, many ticks do end up feeding on hosts like white-footed mice and also chipmunks, which are pretty good hosts for ticks. But we still see ticks in really large numbers on opossums, so it's kind of like, what gives? What are you doing? What are you doing, ticks? You're doing this wrong. You're being stupid. And so that kind of brought me to the idea of like, well, are they choosing their hosts, or are they just kind of going with whatever takes them? And those are two really distinct models of how a creature could select their host. So the passive encounter model is the non-choosy model. So this is the idea that ticks' probability of getting onto a host just scales with the abundance of that host and how common that host is. So maybe mice are super common, and that's why they get on mice a lot, and hosts that are less common, they get on less. And that's just how it is. They're just going through life just waiting for something to happen to them. And whatever's common is going to come get them. The other idea could be maybe ticks are a little choosy to a certain extent, and that explains why we mostly find them on mice, and then we find the few idiots on opossums and other not good hosts. And so this basically says that as a host increase in abundance in an area, a tick's probability of feeding on it maxes out at a certain point. So for a really good host, they might really try to feed on that host no matter what. But for a bad host, it might really need to be super abundant for them to want to feed on it. And I mean, this seems kind of nitpicky, like what does, how much does it matter? We just care that ticks get disease and they transmit it to us. But in terms of the overall dynamic of a Lyme disease system, the method of how a tick chooses its host really matters. So let's walk through that. Okay, so we're going to walk through with this non-choosing model first. Okay, so imagine a population of host-seeking larval ticks and a host community that's dominated by high-quality, high-competency hosts. So these mice are high-quality, they're good for ticks, and they're high-competency, so they're infected and they're able to transmit the infection. And then we maybe have an opossum that's uninfected, and look how cute they are, by the way, look how cute. You expect the resulting nymph population, lots of ticks would feed upon these mice, maybe a couple feed upon the opossums. Lots of ticks survive from feeding on the mice, maybe a couple die from ending up on the opossums. And you get this population of lots of nymphs and lots of them are infected. Counter that with the same method of host seeking, so we have a different population of ticks. We have a different host community. This time this host community has way more low quality, low competency hosts. We wouldn't necessarily have a community where there's only 3 mice and 3 opossums. This is just mimicking like a robust host community where you don't have just mice. If these ticks were to just kind of like evenly feed upon all these different hosts, we would expect a host community where lots of ticks died. Maybe a couple successfully feed, and those that didn't feed, only a few of them were infected. So, in the passive encounter model, decreasing the ratio of high quality, high competency hosts to low quality, low competency hosts will decrease the number of surviving ticks. We love that. Love that. Decrease the infection prevalence in ticks. That's great. That's awesome. And it will decrease the human risk of Lyme disease. That's what we want. Yeah. That's known as a dilution effect, where the host community composition can impact the risk of human diseases in that region. And so let's counter that with this choosy model. So imagine ticks, instead of just going with whatever happens, they have some preference towards these high-quality hosts like mice. So we have this population of host-seeking larvae, host community with high-quality, high-competency hosts. The resulting nymphs, same as before, most of them are feeding upon these mice, they're getting infected, maybe a few still feed upon the opossum and die because they're just unlucky. You know, every group has some idiots, you know. You know how it goes. Okay, now let's imagine the same population where it's more intact, there's fewer high-quality hosts, there's more of these opossums, sweeping up ticks. Remember before, we had a population, more dead ticks, less of them infected. If ticks are instead preferentially feeding upon mice, there would be no difference. It wouldn't really matter. The ticks aren't feeding upon hosts because they're encountered by them. They're feeding upon the hosts that are best for them. So, in a selective exploitation model, by changing the ratios of high quality, high competency hosts with low quality, low competency hosts, we don't see a decreasing number of surviving ticks, hate that. We don't see a decrease in the infection prevalence, we really hate that. And we don't see a decrease in human risk of Lyme disease, awful, that stinks. And so there's no dilution effect, the worst. Okay, so given the consequences of ticks randomly feeding upon the host they encounter versus selectively feeding, like, what are they doing? What should they be doing? How would we know this? So, how do you know that something so tiny just in the environment that you can't really observe its foraging behavior like you can with the lion stalking the gazelle and everything? Imagine, my life would be easier. So I try to pull a bit from other theory, from foraging theory, to get some ideas of what I think we should be doing. So there's this field, so yeah, to what extent are ticks selecting their hosts? there's this field called optimal foraging theory. You don't need to get super into it, but basically it's a field where they do mathematical equations, and they're like, what should something looking for food do? Like, what's the optimal decision to make? And so an example of that would be we have a forager that could feed on a couple different food items. So we have this crappy food item, low quality food item, with certain energetic gain that's lower, handling cost is lower, so each time it's like higher or whatever, it's bad. We don't like it. And then there's high quality food items. So when should the forager just eat the high quality food items? When should it include low quality food items? Well, the forager should only eat lesser quality food items if the energetic gain or the handling cost is greater than energetic gain is greater than you're not even doing that. Visually, what's it look like? Okay. So high quality food items are really abundant. Don't bother with the low quality ones. If you can just easily get those high quality food items, why would you bother with the bad stuff? If you have a pizza right in front of you, why would you go look for a salad? Like, why would you go look? You know, it's right there. Okay. What if high quality food items are rare? Well, then maybe it's worth eating the low quality food items. If the salads are surrounding you, they won't stop. They're everywhere. I guess you eat them. Maybe this is backwards because salads are very nutritious, but like imagine like a bad salad. Okay. So the availability of high quality food items drives choosiness or lack thereof. How does this work with ticks? How does this work with ticks? So this is my overall dissertation question. So for a PhD, you come up with like a dissertation topic, and then you come up with like some subtopics within that to kind of answer that big question. So this is my big question. How do ticks select their hosts? My first question or objective is to determine the number of hosts free-living ticks encounter prior to starvation. That's how often they encounter the pizzas versus the salads. How often are they seeing things? The next objective is to look at how ticks respond to hosts that are known to vary in quality at different stages in the host-taking process. So given that they encounter hosts of different quality, what do? Are they likely to get on them or feed on them, or are they likely to ignore them? This is getting at the idea of do they exhibit choosy behaviors. My third objective is I'm going to smush it all together in an optimality model. And for today, we're just going to talk about the first objective. Because to be honest, I've been working on these first two. Third objective, I'm about like a year and a half, 2 years worth of graduating. Once I finish these two, this is the next goal, to just smush it all together in a math model. And honestly, I don't think it would be fun to present on a mathematical model. Try. Okay, so this is the project I've also been working on here at the Huyck Reserve, determining the number of hosts that free-living ticks encounter prior to starvation. So how do we even do that? How do I determine the number of hosts that free-living ticks encounter prior to starvation? So like one idea could be you measure the abundance of different host species in the environment. And lots of people try to do that as a proxy for encounter rates. Ticks are stationary in the environment, and you have hosts vary in their abundance, maybe that scales. Something to keep in mind though is that animals don't use their environment uniformly. They follow certain paths, so some areas are more likely to be hit more by animals than other areas. So really what you want is you want to just kind of like set up cameras and watch what's happening, see where the animals are, see where the potential hosts are. And because ticks are stationary in the environment, really if you just monitor where hosts are, you can extrapolate how likely a tick in that area is to encounter hosts. And so some questions I have that go with that: do they have fewer or many hosts to choose from? Should they be selective? Does this vary across space, or does this vary geographically? And how does this correlate with things like host density?
Do ticks, when they are looking for a host or find whatever they're doing, do they do it individually or in groups?
Oh, that's a good question. So Ixodes scapularis, they do it more individually. So they are kind of wherever they are parked in the environment, and the typical thing we see is that they climb on up and they start doing this thing, and this is because they have these sensory organs on their front legs and they're just kind of sensing the environment. Other tick species, certain tick species, the larvae are kind of clumped together like a chain of monkeys, parallel monkey situation. So then host comes by, one of the ticks get on, all the other ticks get on. So that's a really interesting question. So in this case, ticks are individually trying to get hosts. Okay, so we talked about how abundance doesn't necessarily scale. We only need to measure host movements. Traditional camera traps aren't really good for what I'm trying to do. So tick hosts include mice and shrews and really tiny things and things like squirrels that move really fast. And people might be familiar with these types of trail cameras, and they're really good. They're really useful. But they're not great in terms of if you want to monitor small mammals reliably, because their sensors don't necessarily reliably trigger with very small things or very fast things, because they're looking at a difference in body temperature between the thing that's moving and the background. And so if you're really small or really fast, it doesn't really pick up on that. Additionally, these things last forever in the field because they're basically in like this like permanent sleep state to a certain extent. And so when it does get triggered, it has a little bit of a boot time. So if something's sprinting by the time this starts recording, you've missed it. So if I want to monitor mouse movement in the field, I would be missing a ton if I tried to use these.
I have to ask you, why can't you set up the camera so that it has sensors that are lateral to the picture taking
So like
camera? So in other words, it gets a notice, there's an animal coming towards the camera, and then it'll take the picture while the
So
animal
you're talking about bringing your own camera system, basically?
I'm thinking about it based on your lecture.
Oh, well, wouldn't you believe? Wouldn't you imagine? I rigged my own camera system. I'm thinking how I'm thinking. So what I did was I adapted a system, and you can't see this from, but I adapted a system from Clemens et al. And I didn't do what you're saying necessarily, but there's these commercially available computer parts that used to be super cheap before the microchip shortage. It used to be $10 per computer board, now it's like 70, so that stinks. But in a couple of years, maybe you can make these yourself, and it's really cheap. And so you get these little computer boards, they're called Raspberry Pis, people might be familiar with them. And you can put this Python program on it, so it's basically a program that tells the camera and computer board to just continuously take pictures of the ground. I have it set up to look at the ground, continuously take pictures, and it's comparing the current frame to the previous frame. And if they're the same, it just discards it, doesn't save it. If there's a threshold difference, it starts recording it onto the SD card as a video. And so I set up 5 of these last year at the Carey Institute, and when I was trying so much stuff. I really thought this project was going to work until I stumbled across this paper. It was the best day of my life. And they're pretty robust. They work pretty good out in the field. I was able to capture so many bugs. So I'm pretty confident that I was catching as many mice as were coming through. I'm using these 940 nanometer infrared lights, which means that most animals can't detect them, so it's not really biasing movement. I think invertebrates, maybe like moths, know what's happening, but...
Wouldn't it be easier just to capture a bunch of rodents and stuff and see how many ticks they have on them?
So that is used as a proxy often to say, well, there's this many ticks on this many hosts, but that doesn't necessarily tell you about the process by which the tick went from in the environment to on the host, because there's other factors that go into that, like host grooming. How many ticks did the host groom off before the ticks that you see on the host? It also gets into, did the ticks, like were there a lot of ticks in one area, or is the host moving a lot? And so really I'm trying to find that initial point of host movement through the environment where they could potentially pick up a tick, but that is a common proxy used in a lot of mathematical modeling. I wanted a direct encounter rate. And so to look at how host encounters vary across space, I have 9 cameras here at the Huyck Preserve off of the Boardway and Racetrack Trail. And I have 9 cameras at the Carey Institute of Ecosystem Studies. And so they're pretty different environments a little bit more piney up here, a little bit more oaky down here. Additionally, at the Carey Institute, I placed the cameras on these long-term mammal trapping grids. So you're talking about looking at ticks on host. I'm putting them somewhere where we are measuring ticks on host and host abundance to see, was that a good proxy measurement? Do we need to go through all the effort I'm going into, or can we just continue doing these mammal tracking programs? Yeah, so this is what mammal tracking looks like. This is a very cute little mouse. Entire time. I actually don't think it has any visible ticks on its ears. I could have chosen a mouse that had a disgusting amount of ticks on it, but I thought people didn't want to see that. This is what it looks like processing them. I wish I could zoom in on that little face there, because it's very cute. Chipmunks, very cool. Okay, so this is what the videos look like. So they're pointing at the ground. Okay, so this might be a little laggy. It's going to start down here, and then move this way. Okay, so this is a white-footed mouse. And we're watching it, and it walks, and it circles, and it's just kind of exploring and using the space and doing its little mouse thing. I have to say, the mice I've been capturing this year, real fat. Like, this was like kind of a normal size mouse. Mice this year. Okay, so that's a video of a mouse. Like, how do we get an encounter rate from that? Like, how do we still get an encounter rate? So we have the raw footage of the path. So this is just a composite image of that video, taking all the still frames and showing this is the path the mouse took in the environment. I can isolate the path, so I'm picking up just these frames. So this is literally just the mouse in the environment. I connect the dots, and then I measure the area of the path. And so it's basically this fraction of red over not red space gives you the proportion of ticks randomly dispersed in this area contacted by the host. So imagine just scattered ticks into this basically square meter. Scattered ticks in there. 2% of ticks would be encountered by this host. Fair enough. That should be accurate.
Okay, so when they encounter it, does that mean they land on it?
Not necessarily. That's the follow-up step to my dissertation. This is just encountered.
Yeah.
Did you get touched by the host? Did you have a chance? Did you have a chance to get on the host?
Or did you reject them?
What?
Or did you reject
Or
them?
did you reject them? Did you have the opportunity to reject them? So I set up 5 of those cameras around the Carey Institute in July and August. So I was really just capturing the larval season, so the larval host-seeking season. And my preliminary results say that the activity of species vary. So this is the number of videos or proportion of videos containing whatever host species of interest. So each dot is one of the camera sites. So some sites had some squirrels happening, some didn't. Some sites had some raccoons really happening and some mice really happening. I only saw one opossum last year. Huge bummer. In terms of cumulative contact probability, so if I take all the path areas of all the mice that went through and all the path areas of all the opossums and raccoons that went through, activity, so the presence of the host and videos, does not translate to contact rates. So you can see that raccoons had pretty high contact rates at some sites. Mice, which were there in pretty high numbers, less so. So do you have any ideas like why contact rates are really different? I'll show this to the audience.
What's the question?
So think about why we're seeing raccoons and mice kind of at similar frequencies on the cameras. But when I measure the encounter probabilities, so those path areas, raccoons are way more likely to be contacted by ticks, or ticks are way more likely to be contacted by raccoons than mice. Yeah.
Because they're fat.
They're fat! The raccoons are big, and they're taking up more space, and they're like taking all the ticks through the environment. Versus mice, like they're common, they're a lot. But they can just dart in and out of this frame, and they're smaller. So yeah, that was kind of cool to think about. This is kind of looking at contact rates between cameras. So each panel is a different camera site. Each color is a different type of species. You can see species vary between cameras. I put birds all into one category. Birds are pretty hard to ID to the species top down. So some sites birds are really common. And all the analyses that follow, I don't include birds because of some other complications I'm dealing with. But there's black bears, raccoons, squirrels. I had a mystery mammal. I have no idea what mammal it is. I don't have a video of it on this talk, but it was weird. So taking those contact rates, compiling them into different sites, you see that contact rates vary between sites. These numbers do not go to the side like I wanted them to be. So probably of contact with a host within a 24-hour period could be 0.2% or 2%, 20%, 0.2. It varies between sites by some pretty strong orders of magnitude. And then if you just look at cumulative daily contact rates times cumulative survival. So here, the average daily contact rate, the average like 24-hour probability of contacting a host is like 1.4. This number isn't perfect. I think there's missing some zeros, so I think it should be a little bit lower than that. But this is the number I have as of my most recent analyses. Times the probability of survival to day X. So larval ticks don't survive super long in the environment. This curve basically says by day 20, less than 50% of ticks are still alive. So if you just multiply daily encounter rate by survival times, you get cumulative contact rates between ticks and hosts. So based on the numbers that I got from my cameras, larval ticks with a mean survival time of 20 days can expect to encounter about 2.7 hosts. So that's measuring the area under the curve. Do we think 2.7 contacts is enough to be really picky?
No. I mean, but I don't understand, because are you saying different mammal hosts, or, I mean, there could be, you know,
We're just looking at-
100 field mice going by,
Mm-hmm.
you know, in 20 days.
We're looking at just that, like you're taking the environment, something's going to brush up on you. We're looking at the probability that something brushes up on you. If you're only going to get brushed up 2.7 times,
Yeah.
do you care what it is, or are you just going to say, this is one of less than 3 chances, I gotta go for it? I think if I was a tick, I would go for it. Keep in mind, this was done last year. Last year was a low mammal year. Who knows what this year's like? I'll know in a few months, but who knows? Maybe it's a bad idea to be. All right, so these are some videos I have from the Huyck Preserve from this year from my cameras. I'll bring back around. So you see that eye glow? This is a mouse. I don't know if you can tell, it's a little fatter than a mouse. This is going to be... Okay, here we go. This is another mouse. Okay. This is something new. It's a squirrel. Really, really fascinating. I'll try to replay that. It's coming in this direction. Real quick. And these are some videos from the Care Institute last year that just shows some of the animals that I did get. So this one was perhaps one of my favorite videos from last year. You can see the absolute sabotage.
Yeah, right. What's all of your cameras?
They're like a meter and a half high, so like 5-ish feet.
Okay.
Look at those little bear booties. So I haven't measured encounter rates between ticks and bears because, as you can kind of see, the idea of measuring contact rate between the grounded bears is a little difficult. This one rascal was really trying to get this camera down. But isn't that fun? This is another good one. That kind of shows you the idea of the surface area raccoons can take up. There's some action that happens in the lower part of the brain. That's it. We'll give it like 5 seconds. 10 seconds, and it doesn't move on.
How much noise do your cameras make when you take a picture?
They don't make any noise.
There it is.
But they do flash red,
They
right?
don't flash anything. So the
That's
lights
a
are
lot
all
of that.
So the light source is at a wavelength that most mammals can't even pick up on. I made sure to get to a wavelength that even mice or nocturnal mammals don't really see, so there's no glow happening. So when
I wasn't referring to that. I mean, the lens,
the lens itself,
when it takes a picture, at least the cameras that I have,
yeah.
red, okay, you see a red dot at the
I don't
lens.
see anything.
You don't?
No.
No noise, no visual difference in the camera when you're looking at it.
When I'm looking at it, yeah, no, I don't see anything.
Okay.
So is this typical of the activity level with raccoons?
It was typical for me last year. So this camera, I believe, was set up near a family of raccoons. And actually, I tried to set up some squirrel traps there for a different project, and I keep getting baby raccoons, so I think it's just a common area for raccoons to play around. And these are juveniles. They're a little smaller, so they're hanging out with each other and being rascals. And then I think this is the last video. This is going to be something coming in from here, guys, if you can see it. Snake? I got a snake! How fun is that?
Very fun.
Yeah. Yeah.
Is it a larger snake?
I can't tell by species.
I know exactly, but there's not many snakes here.
There's not that many snakes. I don't know my snakes super well, but in terms of the snakes I see commonly, garter snake. Yeah. So this is what I've been working on here. Hopefully, by looking at the videos this year, I can estimate robust encounter rates between ticks and hosts and see should ticks be selective. I have follow-up experiments going on that I haven't gotten into today. That's looking at when confronted with a host, how choosy do ticks seem to act? And spoiler, they don't seem to be acting very choosy. So it seems like that passive encounter model that I went through of ticks just maybe feeding on whatever they come by or attempting that could be the model that's happening, and we could be getting true dilution effects depending on the host community composition. So I guess with that, take any questions? Yeah.
It seems to me, correct me if I'm wrong, that you're assuming that all ticks of a species share a common behavior when it comes to selecting.
Right.
And my observation of wildlife is that there's a range of behavior,
Right.
and that's one of the reasons evolution works, is you get success over on this end, but
Right.
not on this end, okay, of the range.
Right.
Now, have you tested to see, I'm assuming you haven't, but have you tested to see if there's variability in the tick population in terms of what they're
individual
doing?
level differences. So honestly, up until recently, most people tend to think of things like ticks as just having kind of like, especially like smaller invertebrates, like some sort of mean behavior expression.
Okay.
And they tend to be centered around that mean. We tend to think that arthropods try to do the same thing. I've been discussing with my PI that it seems like sometimes ticks might have consistently different strategies in different realms. So I spend a lot of time just looking at ticks. I feel like there might be consistent behavioral differences where some ticks spend a lot more time questing and moving around versus other ticks will conserve their energy. And so I think you might be right that there's a range of expression that ticks are having, but you would still expect there to be some sort of mean phenotype. And I'm trying to see where that mean phenotype, behavioral phenotype is landing. And it seems to me that it's landing in a not choosy area. Because we want to know on average, like, what are ticks doing? So look at, on average, what's the disease consequences of what ticks are doing. But it is interesting to note that even really small things maybe have personalities.
Yeah.
Consistent behaviors. Yes.
When you were analyzing how much area a certain animal covered in the like 1 meter square,
Yeah.
how much of that are you using just like an automated program to do?
Yeah. So let me see if I can do this for you back. So I use a program called J, which is this image analysis software that a lot of people use for microscopy. So if you're staining cells and you want to know how many cells are stained a certain color, okay. Yes, so here. So I take the videos and I'm able to export videos into frame by frames. When you import that into J, basically you just click a few different options and it'll say composite image, subtract the background. So I'm able to take a photo with no host, subtract it from this photo, and then you're left isolating the path. So it's fairly well automated. The part that's not well automated is sorting through the videos. So for that, I had a lot of help from an undergraduate named Kendall, who would kind of go through the videos and mark them as there's an animal here, there's no animal here. There are programs that can try to automate that, but it is difficult with the orientation I have and false positives from moths being really close to the camera versus a mouse being very far away. But yeah, if you haven't used J or anything, it's kind of good. And it's free, so we love free software.
You said that you were selecting, or the camera was selecting which photographs to keep
Yeah.
based on changes in the comparison of the previous photograph, right?
Yes.
Now, when the wind blows, these leaves move.
Oh yeah.
Doesn't that create a
I
different?
have a lot of false positives.
You have a lot of them,
Yeah,
okay.
yeah. So that goes into the sorting thing. So I would say, oh gosh, 80% to maybe even 90% of videos at some spot is just based on shadows moving. It's easier at nighttime because there's less shadows happening. But if it's raining, you get the raindrops and the glare and that kind of stuff.
Snow, yeah.
So you have to sort through to see where the hosts are. But when you do have a host, it is pretty distinct from the false positives. Yes.
Okay, I have a few questions. So in your footage, how do you know that it's a white-footed mouse and not a deer mouse? Because they're really similar.
They're very similar. In
It's
general,
It's an assumption that, we do know that they can pass through white-footed mouse.
right. Well, I think in general, the area that I'm at the Carey Institute, you're really just seeing white-footed mouse in the areas where I'm putting these cameras up. So we have those long-term mammal tracking programs. White-footed mice and deer mice, they have very similar behaviors. We expect to find them in the same place. I think they're also potentially interbreeding, so at a certain point, they're not super distinct as species, and they're not super distinct in their role in the ecological system.
What about at height?
What about what?
At height preserve?
At height preserve? We don't know. I assume white-footed mice, and you can kind of go based on, sometimes there's coloration differences that you can see in terms of the fur. I can zoom in and see what that looks like.
Another question. You might have covered this. So I'm sorry if you did. But why is the incidence of Lyme disease and ticks in general so less in West USA?
And then the western, so they don't have Ixodes scapularis. So the Lyme disease system, they do get Lyme disease out on the west coast. The tick that's vectoring it is Ixodes pacificus. So it's a related species to Exodus cabularis. It's called the western black-legged tick. The host community out west and its similar down south, ticks are primarily feeding on lizards. Lizards are not really good host species for the pathogen that causes Lyme disease. And in fact, there's evidence that ticks that were maybe previously infected with Lyme disease that then feed on a lizard, as they're feeding on the lizard, there's components in the lizard blood that neutralizes the bacterium. So then they don't have the Lyme disease anymore.
It's something similar to birds. It seemed like birds were
Birds are common hosts. Birds can transmit pathogens. They can be reservoir hosts. We tend to focus on small mammals. Part of it is potentially sampling bias. So we have these mammal tracking programs. You don't necessarily also have alongside bird banding programs. The Care Institute did do that. They included all these different hosts. And so birds can be pretty good hosts for these different pathogens. And that's actually partially what I worked on. My internship in Southern Maine was looking at anaplasma prevalence in ticks that have fed on birds. And they did have anaplasma.
Okay, this might be a little dumb, but okay, so if you did an experiment where you put a tick equidistant from, say, like a white-footed mouse and like squirrel and possum, how does it decide which direction to go? Like, does it decide?
So there's been experiments like that where they put ticks in the middle of an arena and then they put different host species and then they try to see where the ticks will crawl to, and they use that as an indication of ticks deciding. There are some consistent behavioral differences you'll see with that where more ticks will approach certain hosts over others. In the environment, ticks aren't getting approached by multiple hosts simultaneously, and the hosts are waiting, and the ticks can decide, left is right, and the ticks can buy that, and they're like, okay, that seems like this is a good host to go over here. Ticks are just kind of there, and the idea being that a host will come by, and the tick can decide to get on or not get on. The behavioral experiments I have set up, that's part of my second dissertation question, is looking at how ticks respond to hosts when hosts are just near them. So are they more active? Do they seem like they want to get onto a host? I look at if ticks are moving and a host brushes by, do more ticks get onto the host versus they don't get onto the host if they make contact? And then I'm going to be looking at a data set where they put ticks onto restrained hosts so the host can't groom, and then see how many ticks bit the host and tried to feed versus how many ticks didn't even try to bite the host. So that's going to be the steps of how I look at behaviorally if ticks are choosing to feed.
But in the arena, they have the ability to choose.
If you did the arena experiment, they would have the ability to choose different hosts, but that's not how they work in the environment.
But they're able to.
Are they able to rationalize?
Are they able to rationalize? So ticks, they can sense things. So they have sensory organs where they can sense chemical cues. So they can sense the chemical cues of different hosts, and they can potentially differentiate between hosts. That's kind of what those experiments show. They can sense humidity gradients too. So they follow humidity gradients, and we can measure that they will go down into more humid areas to rehydrate if it feels really dry in certain areas. So they do make behavioral decisions. They're not just like static specks of like nothing. They do things. It's just to what extent can and should they do things. Yes.
I've been under the impression that most of the ticks that come on me are on vegetation above me when I walk by, and they drop.
Yeah.
Okay. Now, do you know if that's a reasonable explanation for how they're attacking humans?
So ticks typically are in the leaf litter. They don't really climb that high. The adults maybe get higher, and they'll climb vegetation a little bit higher. In general, ticks don't want to leave the kind of humid, protective leaf litter. They're not falling on our heads. If you wear a hat, you're not protecting yourself. You can still find ticks higher up on your body though, because they'll enter through your pants or onto your body, or maybe they'll brush your arm if you're sitting down, if they're that high, and they do crawl. So they will crawl up. They're looking for warm spots to bite. So typically they go for areas that are like your armpits, like near like waistbands, any sort of warm protected crevice. And so that can include like neck, in just places.
Do
that
they
are-
hop off a deer?
They won't necessarily hop on one host together. So ticks, feeding is really scary for blood feeding arthropods. Like if you think about a mosquito, you notice it, you smash it. So like once it contacts a host, it's at risk. Ticks, what they do is they fight the host. They inject proteins into the host so that the host can't recognize it, the immune system can't recognize it. It also secretes this like glue, this cement, so that it's really stuck in your body. And so once a tick starts feeding, it can't stop feeding. It can't just decide, oh, now we're gonna bail, now I'm gonna, like, it seems bad, I'm gonna try a different host. They are in it until they decide that they don't wanna feed anymore. And so when they don't wanna feed anymore, they stop producing the anti-inflammatory compounds and they let the host immune system push it out on its own. So ticks can't jump between hosts once they've started feeding. They might drop off hosts if they've never even started the feeding process, if they can't successfully feed on that host, or they don't want to, but they won't be like on your dog and then you're walking like 5 feet away, it won't like launch into space. They're not really strong enough to do that. That'd be awful.
That's why you have to use tweezers to
You
get
have to use
them.
tweezers, you have to, if you've ever pulled a tick off you, you have to use some force. They're in there. They want to stay because they need to feed for like 2 or 3 days.
So do you have an explanation of why when you take a tick off, even if it hasn't engorged or fed very much, why it's still, why that area is still irritated for a very long time?
I think it's probably going to vary person to person based on your own, just like inflammatory response to things in general. Like if you're really responsive to any sort of insect bite or sting, you'll probably notice it more. It could be that it was just enough time for it to irritate your skin. I mean, it's injecting different things. It is making an open wound in the skin. That's enough to irritate you by itself. And it doesn't necessarily need to feed the full amount of time to give you a pathogen. If it feeds for a certain number of hours, it's likely to give you a pathogen. Like I've recently got Lyme disease. There's no way a tick was on me for more than 24 hours because I do pretty in-depth tick checks every night because I'm super at risk. So I think 12 hours was potentially long enough to get a pathogen.
Thank you.